A structural determination of group A streptococcal surface, the hydrogenase, and characterization of its interaction with urokinase type plasminogen activator receptor by Shara Montoya Barreneche. Introduction. Streptococcus pyogenes. The Streptococcus family includes many species of gram-positive coccus, being some of them important for human illnesses. Streptococcus pyogenes, which means pus producer, is a facultative anaerobic microorganism that have fermentative metabolism. They lack catalase, which differentiates them from Staphylococcus, and they can be typically found in pairs or short chains. Streptococcus with clinical value are classified according to the polysaccharide antigens in their wall. In this case, the pyogenes is identified as an A group Streptococcus, or GAS. The human being is the only natural reservoir, and the microorganism cause infections in the throat, the genital tract, the rectum, the skin, and underlying soft tissue. Plasminogen. Plasmin is a broad-spectrum serine protease that degrades fibrin, extracellular matrices, and connective tissue. It's a secreted blood cymogen that is activated by proteolysis and converted to plasmin and angiostatin. Plasmin dissolves fibrin in blood clots, and it's an important protease in many other cellular processes, while angiostatin inhibits angiogenesis. Relation between plasminogen and streptococcus. A large number of pathogens express plasminogen receptor, which immobilize plasminogen on the bacterial surface. Surface-bound plasminogen is then activated by plasminogen activators to plasmin through limited proteolysis, thus triggering the development of a proteolytic surface on the bacteria and eventually assisting the spread of it. Plasminogen receptors and activators are expressed by group A, C, and G streptococci. GAS interact with plasminogen either by direct binding with specific surface proteins or indirectly by sequential binding in fibrogen and plasminogen. Objective. They prepare recombinant SDH tetramer. After purification and crystallization, they determine its crystal structure and characterize its interaction with the extracellular domain of UPAR, referred as soluble UPAR or SUPAR, through 3 canonical assays. SDS pitch, the separation of macromolecules in an electric field, is called electrophoresis. Use a discontinued polacrylamide gel as support medium and sodium dodecyl sulfate to denature the proteins. The method is called sodium dodecyl sulfate polacrylamide gel electrophoresis, or SDS page, and is one of the most widely used laboratory methods to separate biological macromolecules, such as protein and nucleic acid, according to their electrophoresis mobility, which is a function of the length, conformation, and charge of the molecule. Polacrylamide gels restrain larger molecules from migrating as fast as smaller molecules, because the charge to mass ratio is nearly the same among SDS denatured polypeptides. The final separation of proteins is dependent almost entirely on the difference in relative molecular mass of polypeptides. In a gel of uniform density, the relative migration distance of protein is negatively proportional to the length of its mass. If protein of no mass are run simultaneously with the nodes, the relationship between RF and mass can be plotted, and the masses of no proteins estimated. Protein separation by SDS page can be used to estimate relative molecular mass, to determine the relative abundance of major proteins in a sample, and to determine the distribution of proteins among fractions. Recombinant DNA is made by combining DNA from 2 or more sources. In practice, the process often involves combining the DNA of different organisms. The process depends on the ability to cut and rejoin DNA molecules at points which are identified by specific sequences of nucleotide bases called restriction sites. DNA fragments are cut off their normal position in the chromosomes using restriction enzymes, and then inserted into other chromosomes or DNA molecules using enzymes called ligases to produce new genetic combinations that are of value to science, medicine, agriculture, and industry. This describes the process of copying fragments of DNA, which can then be used for many different purposes, such as GM crops or finding a cure for disease. There are two types of gene cloning: in vivo, which involves the use of restriction enzymes and ligases using vectors and cloning the fragments into host cells. The other type is in vitro, which is using the polymerase chain reaction or PCR method to create copies of fragments of DNA. Recombinant DNA technology has made it possible to isolate one gene or any other segment of DNA, enable researchers to determine its nucleotide sequence, study its transcript mutated in highly specific ways, and reinsert the modified sequence into a living organism. In practice, the procedure is carried out by inserting a DNA fragment into a small DNA molecule and then allowing this molecule to replicate inside a simple living cell, such as bacterium. The small replication molecule is called a DNA vector. The most commonly used vectors are plasmids, viruses, and just cells. They are small enough to be conveniently manipulated experimentally, and furthermore, they will carry extra DNA that is spliced into them. Results. Figure 1. According to previous study, the recombinant streptococcal surface dehydrogenase was produced in Escherichia coli BL21 cells. After purification through affinity and size exclusion chromatography, the protein achieved to high purity, near to 99%, as the H was fractioned at 12.5 milliliters on the calibrated size exclusion column, corresponding to approximately 150 kilodaltons, while its bands located near 37 kilodaltons on SDS page. Based on these results, purified SDH was obtained as tetramers, which was consistent with other reports. Figure 4. High affinities yield a stable complex in vitro and can be obtained by gel filtration chromatography. The results showed that there were no complexes formed in the mixture, as each protein was separately eluted. SUPAR was not observed on native page, probably due to its glycosylation. In the mixture, there was no band of the complex compared to each single protein. Moreover, although SUPAR was associated with ATF coupled column, SDH wasn't eluded with SUPAR in the meantime, suggesting that there was no strong interaction between them. Discussion. F. Blasi and P. Carmeliet say UPAR, also known as urokinase receptor or CD87, is a multi-domain glycoprotein and anchored on cell membranes via GPI. It is an essential member in the plasminogen activation system. This was confirmed in the results. Then B. Pancioli and G.S. Catwall say the multifunctional SDH actually contributes to GAS infection and survival in host cells. This was too confirmed in the results. And H. Jean, G.P. Sung, G. Volk, J. Kochar, and B. Pacioli say SDH interacted with host urokinase type plasminogen activator receptor, which mediate GAS infection into human pharyngeal cells. This wasn't confirmed because the union between SDH and UPAR was very fragile. Conclusions. Bearing in mind that the Streptococcus pyogenes affects many tissues in the human body, finding their weakness involves finding the solution or treatment of the illness that the bacteria causes. Also, SDH is very important enzyme in the metabolisms of this kind of bacteria, and that's why it's considered an important target in this investigation. Unfortunately, it's necessary to deepen the studies even more to find the real important in these cells, how it's involved in infection, and how to fight it.